For the last 50 years, scientists have faced a big problem when it comes to functional peptides. It's not so much about the peptides, but rather about the host environment in which the peptides are supposed to carry out their function. Getting a peptide from point A to point B intact, fully functional, in any model system or host can be a very difficult task. This is called the peptide delivery problem, and it's been challenging research for decades. How do we get these fragile peptides where they need to go? Today, I want to tell you about an answer that sounds like it came straight out of science fiction. My name is Dr. Adam Watson, and I'm here at Umbrella Labs to talk to you about the ethosomal peptide research. But first, let me set the stage. Peptides are short chains of amino acids, and they're incredible biological actors. They can regulate hormones, fight bacteria and viruses, activate immune systems, and heal wounds. Nature uses them as its molecular language. But here's the catch. Because they're water-loving and often charged molecules, they can't easily cross a cell membrane. It's like they're locked outside a door with no key. For decades, researchers only had a few options. Inject them directly into a model system or host, which isn't always practical, or we could modify the peptide itself, which often meant losing what made it special because we effectively changed its identity. Scientists needed a better solution. They needed a vessel, a vehicle. Enter nano ethosomes, which are basically tiny, ultra-flexible lipid vesicles. Imagine a microscopic bubble made of lipids, the same stuff that makes up cell membranes, but with a twist. They contain a high concentration of ethanol. That alcohol content makes them remarkably permeable. They can squeeze through tight spaces, penetrate deeply across tissue barriers, and this is the key part, they can fuse with cell membranes. So instead of handing your peptide a map to the cell, we wrap it in an ethosome. The ethosome becomes its Trojan horse. It carries the peptide to the cell membrane, fuses with it, and suddenly your peptide cargo is inside, exactly where it needs to be. The beauty is simplicity. You're not modifying the peptide. You're not using an invasive delivery method like an injection. You're using a delivery system that mimics biology itself. Now, you might be thinking, okay, that's neat, but what can we actually do with this? The preclinical research applications are remarkable. In dermatology model systems, ethosomes penetrate the skin barrier beautifully, which is why they're being used and researched for anti-aging peptides and wound healing factors. Topical application, no needles, better shelf stability, higher efficacy. In metabolic disease model systems, peptide hormones that regulate metabolism, similar to the kind being researched for obesity, can be delivered via ethosomes, opening doors to transdermal administration via creams and serums. In neurology model systems, here's where it gets really exciting. The blood-brain barrier is one of the most selective gatekeepers in any organism. It keeps out everything. However, nanoethosomes can unlock the door. Scientists are now exploring neuropeptide delivery in models of Alzheimer's, Parkinson's, and other neurological conditions. What all these applications have in common is that they were previously limited by delivery. The science was right, the peptides were right, but getting them to work in the host environment was the bottleneck. Why am I telling you this? Because this is what biotechnology looks like when it works. It's not just better compounds or stronger peptides. Sometimes it's about elegance. It's about finding a system so aligned with how biology actually works that it becomes almost invisible in its effectiveness. Nanoethosomes represent a paradigm shift. Instead of fighting biology, we're using it. We're leveraging the fact that cells understand lipids, understand membrane dynamics. We're giving them a message they already know how to read. And here's the kicker. We're just getting started. Researchers are now engineering nanoethosomes to be even more selective, to target specific cell types in specific tissues, to include imaging agents so we can watch them work in real time. Scientists are even exploring combination ethosomes that deliver multiple peptides simultaneously in a variety of experimental models. Because the future of peptides isn't just about better cargo, it's about smarter delivery, and that makes everything more exciting. To learn more, check out all the information on umbrellalabs.is. Thanks for watching.